So, there is actually another paper that which have been reported some result that is different from this paper. That paper find that DEF16 mutants, the lifespan of DEF16 mutants actually increased by H and S E. coli mutants, which is totally different from this paper. So they are looking for the method. That method is different here. In that study, the lifespan assay was performed without FUDR, which is an inhibitor of the DNA synthesis. So then they continue to check whether FUDR is suppressing the activation of the UPR by HNS, which result in lifespan extensions in DEF16 mutants. So from figures 3E to F, you can see that FUDR actually reduces expressions of HSP UPR reporter on both wild-type E. coli and HNS mutant compared to the normal FUDR trick control. So, and also CA was reported to increase the expression of the HSP signal. HNS mutants enhance HSP expressions in combination with UPR inducer MRPS-5 RNA interference from figure 3G. And again, FUDR significantly reduced HSP expression on HNS HT115 E. coli that have been MRPS-5 RNA interference. And this part results summarize that FUDR suppress UPR activity. And next, they also continue to check whether FUDR is affecting the lifespan of the long Lee mitochondrial mutants. As expected, FUDR significantly reduced lifespan of the long-lived mitochondrial mutant without affecting the lifespan of the N2 wild type 1. So we could say that this result suggests that FUDR, at least in part, inhibits C. elegans mitochondrial longevity pathway, such as UPR, which result in reduction of the lifespan of long-lived mitochondrial mutants. And from figure 3I, accordingly, without FUDR, they find that H and S E. coli extend the lifespan of DEF16 mutants, which is likely due to the intact UPR activations. Finally, to confirm that H and S E. coli mutants activate both DEF16 and UPR in parallel to prolong longevity, they generate DEF16 and ATFS-1 mutants in which that both DEF16 and UPR were damaged. And they find that H and S E. coli mutants extend the lifespan of the DEF16 mutants, and ATFS-1 mutants, but the lifespan extension by the HNS mutations was completely lost in the double mutants of the DEF16 and ATFS-1 double mutants in the absence of the FUDR. So taken together from this figure, this result suggests that HNS mutants manipulate the lifespan through CA-UPR-dependent mechanisms, as well as CA-independent, DEF16-dependent mechanism. Yeah. To be brief, that the take-home message here is because as HNS activates UPR or DEF16. So DEF16 is not the only pathway here. We can say that. And this UPR is parallel pathway. So next figure, we come into dissecting the pathway upstream of DEF16. So to identify the molecular mechanism underlying HNS E. coli mediate activation of the DEF16, they aim to determine that whether IIS pathway is involved, because it's actually a pathway that inhibited DEF16. So they measure many of the IIS mutants by knockout and also knockdown with RNA interference, including DEF2, PDK, AGE, and AKT. So, and also they're using RNA to knock down PDK-1. So since that AKT and AK2 played some function there, AKT2 was knocked down in AKT mutants using RNA, and surprisingly that HNS further increased the lifespan of all tested IS C. elegans mutants, which means that IS is totally not involved. So there might be something upstream instead of IIS pathway to activate DEF16 there. And to further confirm that HNS E. coli modulates DEF16 activity independent of the IIS, the DEF16 S3D transgenic C. elegans was generated, in which that all three SER and DHR was mutated to ASP. So all of the isoform was U. And they, as it expected, that the lifespan extension by DEF2 RNA was significantly damaged in DEF16 knockout. This DEF16 S3D worms compared to the DEF16 knockout, only DEF16 knockout. And surprisingly that they found that HNS still prolong the longevity of DEF16 and DEF16 only and DEF16 ST animals compared to the control. So we could say that, and also in contrast to the enhanced nuclear translocations of DEF16 by DEF2 RNA, it was found that HNS E. coli did not induce DEF16 nuclear translocations from figure 4G. So this finding totally, we could say that we could confirmed that H and S enhance deficit transcription activity in the nucleus rather than by affecting the nuclear translocations. And there's many other regulators that also activate deficits like AAK2, JNK1, and SMK1, but they find that there might be some different pathway also involved because the lifespan assay of JNK and SMK AAK2 is not supporting this statement here. So and they next ask whether TORC2, also known as PDK2, was involved in the lifespan regulations. And RIP1, RICT1, is essential components of the TORC2. RIP1 mutants is having a shorter lifespan than wild-type N2 on the E. coli B strand of B50. And where E. coli K12 strand, it extends the RIP1 lifespan compared to the N2. It's from figure 4I here. And from figure 4J, that we could see that H and S did not further extend the lifespan of the RIP1 mutants compared to control. And the long lifespan of the RIP1 was totally dependent on the deficit. And this data summarize that deficit already activated in RIP1 mutants, which lead to no further lifespan extension by H and S. So TORC2 is known to phosphorate the serines on the hydrophobic motif of the AKT1, AKT2, and SGK1. But the AKT family is not implicated in the lifespan extension by H and S, so they next ask that whether SGK1 is acting the downstream of the TORC2 in responding to the H and S. For from figure 4K, that we can see that similar to RIP1 mutants, the SGK1 mutants live longer than N2 animals on the control, E. coli K12. But the lifespan of the SGK mutants was not further extended by H and S. And also from figure 4K that we can see, in addition, deficit knockout suppressed the lifespan extension by SGK. So one more thing to be confirmed, that is involvement of TORC2 SGK1 in the lifespan extension by H and S E. coli. So consistent to the mutant lifespan data, the lifespan modulation by H and S E. coli was significantly reduced in the SGK spectrum. So we could say that SGK1 is dependent on deficit, which is acting downstream, and SGK1 could block deficit effect. And NHS actually enhance the deficit effect. So SGK1 block NHS. That is the take-home. So are we coming to dissecting the components of the MG. So to be sure that Mg is actually the metabolite produced from bacteria to humans, and previous studies have shown that Mg actually activates TORC2, which is involved in the lifespan modulations by the HNS. So, and the screening that in this paper has identified 2 E. coli mutants of Mg degrading enzyme as anti-longevity mutants. So, there's 2 anti-longevity mutants, which is GLOA and YDJG, encoding the enzyme here, which is degrading Mg. So they hypothesized that Mg level might be lower in HNS, which could inhibit TORC2 SGK1 to activate deaf system, and then extend the lifespan of C. elegans. So to test this, the Mg level was measured in HNS using HPLC, and surprisingly that Mg is very low level in HNS. So they also generate GLOA and YDJG over expressing E. coli from figure 5b, and we can see that as expected, this bacteria produce less Mg and extend the lifespan of C. elegans compared to the control E. coli. They're having the control better. So continually compare with the control GLOA and YDJG overexpressions in the HNS background did not further extend the lifespan of C. elegans, demonstrating that the HNS mutation and Mg degrading enzymes is acting through a common mechanism, so they are acting similarly. And from figure 5d, we can see that they find the lifespan extension by HNS was completely suppressed by Mg's supplements compared to the control. So taken together here, this data shows that bacteria-derived Mg is critical for lifespan modulation by HNS E. coli. So Mg is blocking HNS to block the deaf system activity. And from figure 5e, we can see that as with HNS E. coli, overexpression of the GLOA and YDJG in E. coli did not extend the lifespan of the deaf system knockout mutants here. And figure 5e, F. And moreover, the high Mg producing bacteria such as GLOA and YDJG mutants shorten the lifespan of the N2 worms, but not of the deaf system knockout. So indicating that Mg is required deaf system to shorten the lifespan. And next, from figure 5G, there's some recent report that suggests that C. elegans GLOD4 has reduced lifespan, which is a C. elegans genes here. This suggests that protection against Mg is critical for normal aging process. However, there is no previous study that investigated the role of Mg from different sources. So to assess the role of bacteria-derived Mg on the modulation of the host longevity, they grow these mutants on H and S, and they find that they expect that H and S could recover the short lifespan of the GLD4 mutants. However, the GLD4 mutants still reduce longevity compared to the N2 H and S. So this means that the bacteria Mg and the Mg produced by the bacteria and also the Mg within the C. elegans bodies both can modulate the host longevity here. So coming to the mammalian cells, from figure 5H, we can see that the human gut microbe produce a large number of Mg, that the aim to determine whether Mg can modulate the aging process in mammalian cells using HDF. And Mg has been reported that Mg has also been reported to phosphorate AKT at the HM through TRC2. But whether SGK1 phosphorylation is also induced by Mg is unknown here. So they first test whether Mg phosphorates SGK1 in HDF cells, and they find that Mg actually enhances the phosphorylations here. And this suggesting that is from figure 5H. This suggesting that Mg activate both AKT and SGK1 and in turn inhibit fossil in mammalian cells. So from figure 5I, we can see that Mg also induces expression of the synthesis related genes P2221 in a dose-dependent manner. And from figure 5K, we could say that cellular synthesis was induced on the Mg treatment, as shown by the reduced cell proliferations from figure 5J, and a really high level of the synthesis associated beta-galactosidase in HDF cells from figure 5K. Given together that it summarizes that inhibition of the fossil has been shown to accelerate cellular synthesis, and actually Mg is playing an important role there to modulate aging process. And next,